new faces that I recognize here today. Very few new faces, but there are enough, so I have to explain the logo. And those of you who have heard this many, many times, just relax because the best is yet to come. So, do you know what this is? Huh? What is it? Come on, Diane. What? The Brooklyn Bridge. Now, it's the most famous bridge in the world. The most famous. It's the first suspension bridge. And it's the logo for this because it illustrates what the purpose of this course is, which sort of bridges the advances in basic biological and engineering sciences with huge problems in human health. And that is New York on the other side. You're over here in Brooklyn. And we are the people standing on the catwalk. And we're going to be treated to an extraordinary experience this afternoon in discussing, and we hope you'll engage in the discussion, several major issues which we don't discuss certainly every day or with great frequency, even here at the United States. The other important thing in the logo is that life is never the same on either side of a bridge once it's built. The farmers in Brooklyn thought the New Yorkers were going to cause deterioration of the neighborhood, and the people in New York thought the farmers coming across would do the same. Instead, we have two vibrant, incredible cities filled with all sorts of stuff. I won't belabor the point. Now, so Bruce Ames, who was a friend and a leading microbiologist, I think had a talk given here at the NIH quite a few years ago, made this comment that the battle on the surface of a cabbage is more violent than a war between men. Now, of course, what he was talking about are all the things that live on the surface of the cabbage and how the cabbage survives in the face of Lord knows how many different kinds of bacteria, viruses, fungi, metals, you name it. The environment of a cabbage. And so the cabbage has defense mechanisms against it. And this was in an era when the discovery of drug-resistant transporters, proteins, mechanisms was really in its earliest. So in thinking about today's topic, a couple of thoughts came to my mind, which I'll just throw up here, and maybe for those who don't think or about this particular topic very much, you have your own ideas. At any rate, today we're going to have a discussion of wildlife diseases, natural product, and global warming. And so, point one, all living things have evolved and sustained chemical mechanisms to protect against predators, and sometimes those things are what are necessary for their own survival. And biology is filled with this. Even most recently, I read that a scientist at MIT had discovered that the roundworm actually secretes a bile acid, which is what protects it against predators. Who would have ever dreamed that? At any rate, it goes on in many different species. The other important generalization is that many, and I'm not qualified to say most, but I suspect so, but many effective clinical drugs in cancer, infectious disease, think antibiotic, and others are derived from natural products, many different sources. Now, these sources can be plant, they can be microbial, they can be et cetera. And then a big question came to my mind, and maybe we'll have some consideration of this. In the genomic silicone imaging era, where drugs and structures are put together with the aid of a computer, have we reached the end of the line when it comes to natural product searching in the natural environment? And we're going to hear from an expert about this. Now, interspecies disease including us. They're transmitted by viruses, bacteria, fungi, and they can involve insects and other vectors, all of whom have their complicated lifestyle and requirements, environment, and so forth. So a huge question is, how is the changing world, global temperature, pollution of different kinds, oxygen deprivation, and this applies below the water as well as on the surface. How has this changed these paradigms above? And where do we stand regarding this? So this seemed like a very exciting arena to discuss, and we're very fortunate to have two individuals who have a great deal of knowledge and experience regarding this. So I'll introduce both of them. Our first speaker is going to be Jonathan Sleeman. Jonathan graduated from the University of Cambridge in zoology and veterinary medicine. He eventually came to the United States, I believe, in around 2005, where he was a wildlife veterinarian here next door in Virginia in the Department of Game and Inland Fisheries. Somewhere along the line, he did studies involving the gorillas in Africa and their environmental problems. Maybe we'll hear something about that. And in 2009, John went to the United States Geological Survey, and he's the director of the National Wildlife Health Center there. That's part of the Department of the Interior. Now, I always thought the Geological Survey were the people who just made maps. And when I first met Jonathan in Kazakhstan, of all places, I learned there's a heck of a lot more to it. And we're going to hear about a major part of what's more to it, because as you'll see, that branch of which he is the director plays a major role in wildlife health and in mapping diseases, vectors, et cetera, in wildlife throughout the country and in conjunction with others around the world. Now, our second speaker is David Newman, who is known to many of you. David also comes from the other side. Is that a fair expression? And if I can find my notes here. Excuse me. Okay. So David has a broad training. He has an advanced degree in organic chemistry from the University of Liverpool, a Ph.D. degree in microbial chemistry from the University of Sussex, and then later on acquired a Master of Science degree at Drexel University when he got into information science, which is where some of his work is now. Now, Dr. Newman is well known here at NIH because he was the scientist and chief of the National Products Branch of the NCI at Frederick since 1991 until the time of his, I guess, semi-retirement or whatever you call it. Is that correct? Okay, semi-retirement. And his interest in natural products as a source of pharmacologically active material, both marine, microbial, natural product toxins, which can be developed for therapeutic range, metals, and a whole range of interactions at different levels of biology. So we're going to look forward to both of the presentations, and Jonathan, if you would please begin.
Thank you, Wayne. Thank you very much for that kind invitation. And it's okay.
Yeah, you're welcome.
Yep. It's a pleasure to be here with you this afternoon. It's kind of interesting. When I first met Wayne and Luba and others, it was, as he mentioned, a workshop in Kazakhstan that was on climate change and biodiversity. And of course, they came from the perspective of looking at natural products for drug discovery. Our interest was more in terms of climate change and drivers of emerging disease and impacts on on wildlife and conservation, but it was clear that we had a lot more in common than differences in terms of our interest in biodiversity. And I hope to illustrate some of these things in my presentation today, which is really a focus on the importance of biodiversity to human health. Also, I have to mention another strange coincidence, and Dave, you can mention this, but we first met also in Kazakhstan. It turns out we're both from the same hometown. And not only are we from the same hometown, Wallasey in England, we went to the same primary school. What's of most interest is we went at the same time. This is an exercise in difference in aging. Looks like I fared a little better than you, my friend. Okay. Okay, so what I've got for you this afternoon is I'm going to give you a little sort of overview of the drivers of emerging disease, what are the different environmental factors that are causing these diseases to emerge. I'm going to talk about some of the examples of emerging diseases. I picked three. One is purely a wildlife agricultural disease, but it's a very good illustration of some of the impacts of climate change. This is hemorrhagic disease and blue tongue virus. Probably not something that's very familiar to you. But anyway, then I'm going to talk about avian influenzas, which should be quite familiar to folks here at NIH, and then the bats and the disease. And then to end, I'm going to wrap it up. I'm going to sort of talk a little bit about the One Health approach and perhaps some of the things that we can do to help manage and prevent these things from occurring in the first place. It's kind of interesting. If you look back in the literature in the 1950s and 1960s, there are very few publications on diseases of wildlife origin. It was very much a neglected endeavor scientifically. They weren't really considered important in terms of impacts on wildlife populations. There weren't that many examples of zoonotic diseases, maybe rabies and a few others. It was also around the same time. Now, it's unlikely that he said this quote. Dr. William Stewart was the Surgeon General back in the 60s. But I think it reflects the sentiment at the time with the advent of antibiotics and vaccines that we very much had won the war against infectious diseases, and we should shift our focus to more chronic diseases like cancer, heart disease, and so forth. Now, I live in Madison, Wisconsin, so I'd be remiss not to mention Aldo Leopold. He's from Wisconsin. He's the founder, really, of modern wildlife management here in North America. Wrote Sand County Almanac, just a seminal publication on wildlife conservation. I think he was rather prescient in his understanding of the role of disease in wildlife populations, as well as the importance of the environment being a major factor in causing disease emergence in the first place. So fast forward several decades, and I think we can agree that the book on infectious diseases has largely been dusted off. And we're living in a global environmental situation that's very much favoring the emergence of new and novel diseases, mostly infectious diseases. And what's of particular interest to us is the fact that many of the diseases that are emerging have a wildlife origin or a wildlife connection. I think 70% of the emerging diseases, 60% of the emerging diseases are of animal origin, of which 70% of those are of wildlife connection. Again, I think there's general consensus that these drivers of diseases are largely a result of human activities. And probably the biggest category, and we'll talk about this in the examples, is landscape change. Changing farming systems, bringing wildlife, domestic animals, and humans into closer contact. Encroachment of humans onto wildlife habitat and exposure to species they hadn't been previously exposed to. I'd put climate change in this category. Again, we'll talk about some of the changes in severity and distribution of diseases as a consequence of climate change. And I put a picture of the polar bear up here in climate change. Do you know why? Yeah, so it's the first species to be listed as endangered as a consequence of climate change because of loss of habitat, which is the sea pack ice up in northern Alaska. And it's kind of interesting because these polar bears are now no longer hunting on the sea ice, which their main prey is seals. They've moved inland and they're scavenging hunting on birds and other species, kind of changing their prey base. And we're seeing some health effects as a consequence of this, as well as they're getting into a lot more conflict with people that are living on this land. So it's kind of interesting the ripple effect. Obviously, the sort of human demographic and behavior changes. I think it was 2007 when we tipped over for being mostly rural communities to most people living in an urban environment. So that increasing urbanization is clearly having an impact. There's also the issue or the concern about the constant concern about deliberate release of agents as bioterrorism, weapons. And most of those also have some sort of wildlife connection, maybe plague, tularemia, and botulism. I think for me, the biggest category is global travel and trade. The movement of people, the movement of products has broken down those geographic barriers that used to prevent diseases jumping from one continent to another. And again, we'll see some examples of that. These microbes are biological organisms, so they undergo evolution as a consequence of selective pressures. And so we're seeing changes to their genetic structure and behavior. Probably the biggest example of this is the multi-drug resistant bacteria. Again, there's some predictions that some of the antibiotics common antibiotics will no longer be useful in the next 20 years. But some of these organisms also naturally undergo evolution and change, and the influenzas are probably the best example where they naturally undergo drifts in the genetic structure as well as major reassortments into or so-called shifts of form where they form new different subtypes. And this may seem like an odd category, but lack of infrastructure. If you don't have the surveillance systems, if you don't have the mechanisms to respond and research these diseases, the outbreaks and the consequences tend to be more severe. We see that when we get in wars and famines, where there's breakdown in public health infrastructure. But also, and this is my little soapbox, we really lack the same type of infrastructure that we have for public health and domestic animal health for wildlife health. We don't have that sort of network of laboratories across the nation. We don't really have that capacity to respond quickly to disease outbreaks. And I think as a consequence, we always tend to be behind the game and trying to play catch up when we see these disease issues. And I probably don't need to explain to you the impacts of these diseases, and just several examples. And I wanted to go beyond the public health impacts, but one of the best examples, unfortunately, is HIV, which is now known to be a mutated form of a simian immunodeficiency virus, probably spilled over to people from hunting and butchering of primates in Africa. And they're now estimated to be 35 million people living with HIV infection globally. Also has an impact on food security. There are about 400 million poultry at least have been culled since 2003, 2005 in efforts to control outbreaks of highly pathogenic avian influenza, which has had an impact on food security and economic development of some Southeast Asian countries. And I think increasingly recognized as I probably should use a little mouse, is the biological and ecological consequences of these diseases. They are now causing some pretty marked declines in populations. And some of these have very important ecological roles or provide ecological services. Probably the best example is colony collapse disorder of bees. They're major pollinators. There's now some areas of the world where crops have to be hand pollinated. And here in North America, I don't know how many of you are familiar with bat white nose syndrome, which is causing precipitous declines of bats across the US. A little plug for us. So we still don't really understand what colony collapse disorder is caused by. It may be multifactorial, and USDA is still very much trying to research that. USGS, though, we figured out white nose syndrome within 6 months, so we're much better than the USDA in these types of investigations. Yes, I'm pretty safe that no one from here is from the USDA. This is kind of interesting graphic. Kind of shows the types of diseases that we've investigated since our foundation in the 1970s. And you look at the top line, diseases like avian cholera, avian botulism, they're important wildlife management issues. They can cause a fairly large die-off of a waterfowl on a refuge, but they're not a human disease, they're not an agricultural disease, they don't really have any economic impact. And they remain confined to a specific geographic location and don't really threaten the persistence of wildlife populations. Whereas if you look at the diseases since the 1990s, first of all, you notice the number of new diseases popping up. It seems every 2 or 3 years we've got some new novel disease to address, starting from amphibian diseases in the 1990s, West Nile virus, monkeypox, probably some of you remember that in 2003, chronic wasting disease in 2002, and then more recently avian influenza outbreaks. And right now, actually, kind of interestingly, dealing with 3 different fungal infections: white nose syndrome, snake fungal disease, and this new fungus of salamanders, which we're very concerned about introduction to the US, because the United States has the highest biodiversity, species richness of salamanders in the entire world. I think the consequence of these diseases are far more severe. They're spread very rapidly across large geographic areas. West Nile virus went from detection in New York in 1999 to the West Coast in 2003, within 4 years. They're causing large-scale population declines. With amphibian diseases, even caused extinctions. White nose syndrome, some species now are predicted to go locally extinct in the next 20 years. And they're also increasingly a public health and an agricultural disease. Avian influenza, I think, being the classic example. So I think increasingly we're having to work across these sectors to address these issues of mutual concern. All right, so I'm just going to give you a few examples of these emerging diseases of wildlife origin. And for the examples, I hope to illustrate some of these drivers. I'm going to talk a little bit about sort of the dichotomy of wildlife, in that they're obviously the origin, the source of these emerging diseases, but they're also impacted by these diseases, and how can we kind of balance those two different, almost competing interests. To answer my own question, I hope to illustrate some of the value of biodiversity to human health, and finally talk about some of the One Health solutions, some of the sort of solutions we can look to implement. So climate change. So the Intergovernmental Panel on Climate Change that published a report in 2007 predicted unprecedented rises in global temperatures, rises in sea levels, and changes in precipitation patterns. So it's not just global warming, but it's an entire change to the climatic systems. They also predicted we're going to have significant impacts on animal and human diseases. And we are starting to see, and have seen for a number of years, these particular impacts. They're having some profound impacts on the geographic distribution of diseases, the incidence of disease, and some of the severity of some of these diseases. So we're witnessing changes in geographic range and distribution, diseases at higher latitudes and altitudes where they previously didn't exist. We're seeing changes in animal and plant phenology. So it's the timing of migration and the timing of emergence of different animals and plants. And this is having consequences not just for wildlife conservation, but also in disease transmission, particularly tick-borne diseases. We're seeing some direct effects of these climate change, heat stress in moose in Minnesota, where they evolved to survive in a much colder climate and cannot cope with the increased winter and summer temperatures. We're seeing changes in the virulence and genetic diversity of these organisms, particularly like, for example, West Nile virus. We've seen some new evolution of new strains of West Nile virus that are far more severe, far more pathogenic. And we're seeing some just, again, changes in patterns in these diseases. And Dave's going to talk about some of the impacts of climate change on coral reefs and declines of reef ecosystems. So I mentioned that the disease I wanted to sort of use to illustrate the impacts of climate change is so-called blue tongue and hemorrhagic disease. This is mainly a disease of agricultural production, as also disease of wildlife. It's caused by orbiviruses, and they're transmitted by these little things, Culocordes midges. So it's mostly a disease of late summer, early fall. And in high outbreak years, it causes a very acute disease, mostly in domestic and wild ruminants, especially sheep, goat, and deer. You can see why it gets its name. You get a marked edema, hemorrhagic edema within the lungs and other organs. Causes a cyanosis. The animals can't breathe. You can also see here the edema around the eyelids, and they can die within 24 hours of the development of clinical signs. So it's a pretty rapid disease. The classic presentation is because they get a very high fever, they often congregate in water sources. So you get these deer, these sick, dead deer, and kind of congregation in ponds. You may hear about it every few years. They get some very major outbreaks. Actually quite common here in Virginia or around the DC area. It's also, as I mentioned, a disease of sheep and goats and cattle. And what's interesting is we're starting to see some dramatic extension of the virus northwards both in Europe. So in Europe, it used to be confined to the sort of Mediterranean region. But several years ago, in 2005 in particular, they started getting outbreaks of blue tongue here in Central and Europe, and then into England or Great Britain, and also into Scandinavia. Around the same time, we're starting to see changes in distribution of hemorrhagic disease here in the United States. And so really was confined, actually right here, the sort of Maryland state line. It really wasn't seen much further north than this line. But now we're starting to see outbreaks up here in Wisconsin, up here in New York for the first time in 2007, and starting to spread further west. We're also, intriguingly, and this is somewhat of an aside, starting to see some different serotypes. And these exotic reassorted serotypes. One particular called serotype 6 was found in the US several years ago. Now, if you look at the map here, this is showing the change in average surface temperatures of Europe, the top map in particular. So what you can see is actually Northern Europe, Northern and Central Europe, has increased at a warmer pace than the rest of Europe. And while it's correlative, we believe that this warmer climate is allowing these vectors to survive these higher latitudes, and therefore allowing them to transmit the disease in areas where they previously couldn't survive. These higher in winter summer temperatures also increase what's called vector capacity and competence, the frequency of biting, the viral load in these midges is also higher as a consequence of climate change. There may be higher population levels of these vectors because of increased life cycle of these vectors. So very much strong evidence that climate change is leading to this changes in geographic distribution. Again, on the side, what we believe is the novel stereotypes maybe as a result of importation of exotic animals or viremic cattle from other areas. So again, illustrating that sort of role of travel and trade in emerging diseases. So to kind of just look at this a little bit more detail to try and get a little bit more data, we did a study where we looked at hemorrhagic disease in white-tailed deer here in the United States to see if there was any correlation between incidence of the disease and different climatic factors. So in addition to causing acute disease, it can also cause a chronic infection. And one of the primary signs, I don't know how many people hunt deer in this room. No one. Wow. So, well, if you hunt deer, you're almost certainly seeing this lesion, this interruption in the growth of the hoof. So obviously, when they get the viremia and when they get the fever, it causes lesions in the coronary band here of the hoof and causes a split. And often the hoofs will sometimes slough off. So this and the growth rate of the hoofs is such that it will, if they had hemorrhagic disease one year, by the time you see it next year, it would have grown out. So it actually turns out to be a very good measure of incidence, where we can measure the number of new cases that happen each year. And so we collected data at game check stations, right? We sort of, when hunters bring in their game to have them checked, we were able to measure the percentage of deer that had these hoof lesions. And then we did some statistical analyses to look at the correlations between hemorrhagic disease incidence and different climatic factors, winter temperature, summer temperature, precipitation patterns, and so forth. And what we found, and this is just a graph showing the incidence or the percentage of deer that have this cracked hoof lesion, and this is just the variation over years. And what we found is the years that we find this higher incidence are the years that have higher winter and summer temperatures, as well as the years that have lower rainfall. And so what we believe is obviously, as I mentioned, these higher winter and summer temperatures increase this vector capacity and competence, allowing them to acquire, maintain, and transmit the virus at a much higher rate and much higher viral load, as well as increasing abundance, survival, and biting frequency. But what's interesting is the lower summer rainfall, what that probably is doing is improving or increasing the breeding sites of these midges. So what happens, especially in this part of the world, when you get sort of drought conditions, you don't lose the water completely. You get the receding of the water line. This exposes the mud flats of the beds around these lakes, and that's the prime breeding grounds for these midges. They don't breed actually in the ponds around these kind of muddy flats. And so what we think is happening is it's improving habitat for these animals, for these midges to breed. And there may also be a role of physiological stress on deer during these very hot, dry summers where they actually may be stressed and more susceptible. So I think this is a very good illustration of some of the complexities and some of the consequences of climate change on disease, incidence, and distribution. The next example I wanted to touch upon is avian influenzas. This is probably something a little bit more familiar to this group, but as you probably know, wild birds are the natural reservoirs for all type A influenza viruses. They've been isolated from over 100 different species of wild birds, but mostly from the waterfowl, the water birds, ducks, geese, swans, as well as the shorebirds. You probably know they're classified into neuraminidase and hemagglutinin subtypes based on these surface proteins. They're also classified into low pathogenic forms and highly pathogenic forms based on their ability to cause disease in poultry. And that does not relate to their ability to cause illness in people or in wildlife. And again, I probably don't need to explain to you the public health significance of avian influenzas. They're the progenitors of all the pandemic human viruses that have been identified so far. These are just schematics of the genetic structure of the different pandemic flu. Blue shows the avian genes. And so they all have an avian origin. In fact, it turns out the 1918 Spanish flu was a pure avian influenza virus. They also have large economic costs. As I mentioned earlier about the efforts to control H5N1 in Asia, it's cost up to $12 billion to those economies. I don't know whether you remember last year, there was a large outbreak of highly pathogenic avian influenza here in the United States, in the Midwest. That resulted in about $4 billion in economic losses, both from trade restrictions on poultry products as well as efforts to control the disease. And it seems like Southeast Asia is a major focus for the emergence of these avian influenza viruses, H5N1, H7N9, which is kind of interesting virus because it's low pathogenic to poultry, but causes human disease, which is a very unusual property. H10N8, H5N8, and so forth. And it's highly likely, as I mentioned, it's a farming and market systems particularly in China that are driving the emergence of all these new strains of avian influenza. This is a map we generated showing the distribution of waterfowl populations in China. You can see here in the blue the major concentrations of waterfowl in the southeast of the country, and also mapping here the major poultry farms and poultry industry. And as you can see, unfortunately, they overlap quite nicely. There's a lot of open farming systems in China where they raise these poultry out in these fields in the rice paddies where they can commingle with wild birds and obviously get that viral exchange. And then they transport these chickens to the markets live, where people purchase them and are slaughtered there on the spot, again increasing human exposure. So it's very much this kind of farming and market system that's driving the system. The challenge is these viruses have not just spilled over from waterfowl to poultry, they spill back into wildlife populations. And over time, these birds have evolved to where they can now carry these viruses over large distances. So one of the main projects my center has been involved with is this surveillance for avian influenzas in wild birds. And we've been doing this since 2005. And then actually in 2014, we actually detected a high pathogenic H5N8 in wild birds in North America for the first time. Timing in December coincides very much with migration, not natural migratory patterns. And we were able to demonstrate these waterfowl could be asymptomatic carriers of these viruses. If you look at this map, you can see that the America's Pacific flyway overlaps with the East Asian Australian flyway. So it's likely these birds pick up the viruses during winter in Southeast Asia, and then they commingle with North American species up here in Siberia for breeding. And then they migrate back down south, kind of bringing the viruses back down with them and then causing these outbreaks. And about the same time that we were having an outbreak, South Korea and Japan also suffered some outbreaks detection, both in wild birds and in poultry in their countries. And sure enough, when we did phylogenetic analysis of the H5N8, it found that all 8 RNA segments were greater than 99% similar to Group A 2014 South Korean strains. So not only was this the first time that we found a highly pathogenic virus in wild birds in North America, it's the first time a pure Eurasian strain of virus had been found. And as I mentioned, it went on to spread very rapidly across the country and causing some pretty large poultry outbreaks. You can see here this virus clusters here with the Eurasian strains, not with the North American strains. Actually, most closely related to a sample from a crane. You can see here all the North Korean samples. Just as an aside, it's kind of interesting because we do a lot of wildlife disease outbreak investigations. And in 2012, 2013, there was a large die-off of harbor seals up in New England that we became involved in the investigation. Turns out they died from a H3N8 avian influenza virus, caused a marked infection of the lower respiratory tract. Again, when we did characterization of this virus, it turns out to be very similar to a virus that circulates in North American waterfowl, but had mutations indicating adaptation to mammalian hosts. And as you know, the main theory of how human pandemic viruses evolve is the mixing vessel theory, mixing vessel hypothesis. We get avian viruses and human viruses mixing in an intermediate host like the pig, and that's how these viruses then recombine, reassort into these pandemic forms. But actually, I wonder whether this case illustrates the sort of role of called a direct pipeline of viruses, where this is perhaps more frequent or more common than we perhaps have thought previously or historically. So again, it illustrates the dichotomy of wildlife. A lot of people regard them as villains, the sources of these outbreaks as the sources of human pandemics. But they're also equally impacted on by some of these viruses. Turns out that raptors, birds of prey, are highly susceptible to these avian influenza viruses. It also raises concern, again, in my wildlife background, about the safety of consuming of waterfowl collected from hunting activities. It raises questions about public perception of waterfowl, participation in recreational activities related to waterfowl. So, and after these outbreaks in North America, there was a fair amount of finger pointing. It became quite a controversial issue, the blaming of the wildlife for these outbreaks. And I'm getting a little bit to the sort of conclusion of my presentation, but I kind of found this nice little quote from this publication looking at biodiversity of influenza viruses in wild birds. And the emphasis is mine, and I think rather than pointing fingers, but who's to blame, is let's focus on interventions where we can sort of allow wildlife, humans, and domestic animals to coexist. And for avian influenza, improving biosecurity at the farms and markets probably is the key to prevention. We're not going to be able to eliminate influenzas from wild birds. They're always going to be carriers, but finding those epidemiological routes of transmission and focusing our efforts on that, I think, is going to be the most cost-effective control method. And finally, bats. Bats are absolutely fascinating creatures as far as I'm concerned, but also fascinating from a public health perspective in that they seem to be the source of some of these major emerging viral infections. The coronaviruses, SARS and MERS, the paramyxoviruses like Nipah virus, Hendra virus, the filoviruses, Lissa viruses. So there's a huge diversity of viruses within these species. It's raised a lot of questions about what's unique about bats. Is it just simply their biodiversity? They're the second most biodiverse group of mammals behind rodents. There's about 1,300 species of bats out of about 20,000 species of mammals. Or is there something unique about their immune system that allows them to coexist with these viruses? And so there's a lot of questions being raised about the role of bats. Interestingly enough, we're now doing some research on these diseases to see if we can develop vaccines that can be delivered to these species to vaccinate upstream, rather than how we can prevent the spillover of these viruses by vaccinating bats against these different diseases. Right now, starting with a rabies model or a Lissa virus model, but hopefully be able to extend it to some of these other diseases. But also, tragically, I think, again, bats are equally victims to these emerging diseases. And I think that what makes them unique also makes them prone or susceptible to some of these disease threats. And I think all of you have heard of bat white nose syndrome. We first discovered it actually 2006, 2007. It's characterized by large-scale die-offs. You can probably see the bat carcasses littered here on the cave floor, as well as growth of this white fuzzy material on the muzzles, hence the name, white nose syndrome, but also you can see it on the unfurred or non-furred parts of the body. Turns out this is a fungus. Turns out it's a newly described fungus. It's never been described previously. It's called Pseudogymnoastis destructans. It's also the first time a fungus in this genus has been recorded as a pathogen of a vertebrate. So it's a completely new group of pathogens. Like many times when there's something novel like this, it took a while for the scientific community to accept this was the actual etiology of white nose syndrome. Fungi are very rarely primary pathogens. Athlete's foot is irritating, but doesn't kill you. There are some other examples. So Cryptococcus in people, histoplasma. So anyway, what it turns out, there's any pathologists in the room, is these fungi are not just remains on the superficial layers of the skin. This is a cross-section, histopathological section of the wing of a bat. And you can see how the fungal hyphae are invading deeply into these tissues. So it's a very invasive fungus. Now here's the bonus points. What else do you notice unusual about this histopathological slide? Is there any pathologists in the room? No. Well, if you get this, you're a genius. So there's no inflammation, right? Normally, when you get an invasive in the tissue, you'll see inflammatory cells come in to attack. So either this fungus releases immunosuppressive factors, or these bats, because they're hibernating, their immune system is naturally downregulated or suppressed. So this fungus attacks them in a very vulnerable part of their life cycle. We've done experimental trials to fulfill Koch's postulates. We've proven this is the actual primary cause. We've also done experiments to demonstrate how it actually kills the bats. What happens when you get these severe infections in the integument, you get a lot of water evaporation and loss, as well as salt imbalances, very high hyperkalemia, which can cause physiological disturbances. It also causes them to arouse, burn off their body fat, and they go foraging for insects in the middle of winter when there are no insects around. And so that's why we see these large-scale die-offs. What was kind of interesting with this disease, when we first started publishing our findings, we got contacted by colleagues in Europe. They said, hey, we've seen the same thing in Europe, but we haven't seen the mortality in bats. So they sent us some samples, and we sequenced them, and they were the exact same fungus as what we found in North America, except what we had in North America was a single clonal species. There's a lot more diversity in Europe. So what we believe is this is a European pathogen that somehow, or European fungus that somehow has been brought over into North America. A lot of people ask me why bats in Europe don't suffer the disease, and we still don't really know. There's a couple of hypotheses. One is bats behave, the lot larger bodied species in Europe, they also congregate in smaller groups. So we think they may be more resistant to the fungus. They may have co-evolved with this organism. But you also look at the fossil records of some of these caves. Several thousand years ago, there's a layer where there's a large number of bat bones. So another hypothesis is they somehow underwent a mortality event a long time ago, and these are the remnant populations. And it's certainly true, population density, well, in Europe is a lot lower than it is in North America. We have bats that hibernate like 50 to 100,000 bats in one location, or used to. So that's a couple of theories, but for me, again, it's an illustration of what I think is a major driver of these diseases, is the ability of these pathogens to jump continents, hitchhike or piggyback on people and equipment, products, and so forth. So, and it's had some pretty major consequences. So I think also this is a great illustration, I think, of some of the ecological and economic implications of loss of biodiversity. Bats, as I mentioned, are primary predators of insects. They provide very important ecological services. They're said to be able to consume the body weight in insects every evening. So tons and tons of insects are removed from the environment every night when they emerge from the roosting areas. And they're probably going to have, this loss of bats are going to have ecological repercussions that we probably cannot predict. There may be some health effects, human health effects, increased abundance of vectors of disease as a consequence. We do know there's going to be agricultural and economic impacts. They predate a lot on moths that cause destruction of crops and forest products. And we did a study to show that bats contribute up to $50 billion to the US agricultural economy through their insect control services alone. And obviously, you lose the bats, we're going to see increased use of pesticides and some environmental consequences as a result. So again, I think coming back to the main theme here, it tells me that, illustrates the importance of biodiversity to human health and well-being. So how can we coexist with bats? What's the, the villains? I mean, the source of a lot of emerging diseases, and they're also the victims. And I just want to walk you through this nice little study and this story, because I think it points to perhaps some of the things that we can do in the future. So several years ago, there were outbreaks of Marburg virus at cave environments in Uganda. You may recall the incidents. Actually, tourists was infected as well, some local populations. As I mentioned, fruit bats are thought to be, Egyptian fruit bat is thought to be the major reservoir. So the CDC went in and did a study where they seasonally sampled these bats to look at did PCR testing, look at shedding of Ebola or Marburg virus from these bats. And they found that shedding occurs at 2 major times of the year. This is actually during breeding season where the juveniles emerge. And that's very consistent with what we know about most emerging diseases. The juveniles are usually very susceptible. They have high burdens of the virus and therefore shed the virus in larger amounts. What was interesting is 85% of the human cases occurred during those times of year where we saw major peaks of shedding. Now, the CDC informed the local population of these results. Do you know what they did? They went in and killed all the bats. They decided they couldn't coexist with these bats. And so they went in and extirpated them all. They used the caves for a lot of mining activities and obviously for tourism. Well, there's an interesting sequelae in that since then, the cave has been repopulated by other bats. They've gone back in, they've repeated a similar study, and actually they've found the bats that returned have a higher prevalence of Ebola or Marburg virus than what they did previously. So not only have you not solved the problem, you've made the problem worse. And again, are there some interventions that we could do that allow humans and bats to coexist? Now, maybe with Marburg virus, you don't want to take any risks, but could we allow human access during the non-risky times of year where they could mine and avoid, close off the caves during the high-risk times of year? What are they mining for? And there's some other alternative ways to get those products. They don't need to access the caves for their livelihoods. Perhaps a little sort of outside-the-box thinking is something that we need to do to help look at some of these challenges. Okay, so just to conclude, again, I'd sort of like to pull this together a little bit and talk about the One Health concept. You've probably heard of this, the idea that human, domestic, animal, wildlife health are all interconnected within the context of ecosystem health or environmental health. And the fact that all these drivers that are driving wildlife diseases are also driving human diseases. And also, when we get changes in environmental health or ecosystem health, especially with the loss of biodiversity, it starts to jeopardize these ecosystems upon which all life depends. And so the idea or the definition of One Health, or at least most commonly accepted definition, is collaborative efforts of multiple disciplines working locally, nationally, globally to attain optimal health for people, animals, and the environment. And the emphasis is mine on the optimal. And I think, to me, that's key to implementation or utilization of a One Health approach. Can we take a systems approach to developing these interventions to reduce risk that balance the needs of humans and wildlife? And I just will show you a few kind of illustrations of that. Oops. Some of this we've talked about. You know, rather than blaming wildlife and culling wildlife, focus on increased farm biosecurity. That only prevents these viruses getting into poultry, but also prevents poultry viruses getting into wildlife. I mentioned about spatial and temporal separation of humans and bats to prevent Marburg spillover, but there's other examples, I think, where we can implement that type of intervention. I think there's a lot can be done in risk communication approaches, changing human attitudes. Many of these diseases have some sort of human behavioral component to it. I really like this advertisement or this sort of campaign from EcoHealth Alliance. It was a sort of campaigning against the illegal wildlife trade. But the message is not just this is endangering their lives, it's endangering your life as a consequence of the movement of these pathogens and potential zoonotic disease. Are there some conservation reasons for improved public health and food security? So I was reading on the USAID's website, they got this big emerging pandemic threats program, and one of the campaigns is to prevent the spillover of viruses and bushmeat into people. Their intervention is to provide education and training to hunters on personal protection and hygiene. Well, that may prevent your viruses spilling over, but does nothing to stop the bushmeat trade, right? And so maybe this can be viewed from a food security issue, and are there alternative sources of protein for these communities that can be generated that decreases the demand for bushmeat, prevents these zoonotic diseases from spilling over, but also allows a healthy ecosystem to coexist. And I think, ultimately, and this may seem an obvious message, but focusing the actions on what is the ultimate driving force for these things, I think is going to be the most cost-effective point of control, and alleviating poverty, so there's less depletion of natural resources, less environmental degradation, less exposure to infectious diseases, which then causes further biological depletion, is, to me, I think, again, the common point of interest that we all have in managing these disease issues. And finally, partnerships. Again, I think that's the major message of One Health is we need to develop national, international partnerships and governance structures to address these issues of transboundary concern and build up our capacity. And this is a plug for my field because, we got public health, we've got domestic agricultural animal health, but the wildlife leg is missing, and we very much lack that infrastructure and capacity to address these wildlife diseases. And I think that's a major gap in our holistic approach to management of these disease issues. That's all I have. I'd be happy to questions now or later.
Well, we're running just a little bit late. Maybe we have time for a couple of questions while the topic is immediate. Are there any? So, Jonathan, did I understand that you can identify the mutation in influenza virus in birds which are just beginning their migratory cycle?
Mm-hmm.
Now, I don't know how long it takes for them to fly, but does that mean, what impact does that have on vaccine development, which are specific for the mutations for influenza?
So I think it could have some. So these viruses can remain unchanged in these wild birds over significant differences. So you can see this sudden introduction of a novel strain. So I think that does have some implications in the vaccine strains and the importance of the different vaccine strains. In fact, some of the viruses we isolated have gone, I forget where they go, to actually be part of that suite of viruses they use when they develop vaccines. And I think it also illustrates the importance of why we need to be constantly vigilant, because as I mentioned, these viruses constantly reassort with other strains and develop new properties. And now we've got highly pathogenic avian influenza in wild birds. That's a game changer. That's a whole ecosystem these viruses got to play with that they've not had before. So we may start to see more and more of these strange viruses emerge. So I do think it's, whilst mostly what I focused on is the wildlife agricultural interest, I think there's also potential public health issue.
Any questions now?
Yeah.
Oh, Luba?
I have a quick question.
Sure.
With the emergence of the more recent infectious disease and Zika virus, is there really confirmed that humans are the only species affected by Zika? Is there anything in animal world that looks like Zika virus and the microcephaly or
Yeah.
syndrome?
Yes. So Zika virus is a great example. Now, what we believe thus far is only primates and humans. Primates are the main reservoir, and all humans. There's no evidence thus far that there's a wildlife connection or wildlife reservoir. But interestingly enough, we're starting to do some experimental work to see if birds or other species could potentially be reservoirs. And like anything, when these viruses come to new ecological niches, they often change behavior, and they may find a species that they had never previously before that could be a very competent reservoir. So again, I think we need to be very, very vigilant about these issues. So right now, the answer is no, but we're looking quite closely to see.
Okay.
Great, thank you.
Jonathan.
Appreciate it.
Thank you very much.
Do you need this?
Great.
Thanks, John.
Thank you.
Okay. Right. Now for a slightly different perspective, I should add that I joined the Natural Products branch in 1981, where Gordon was my chief. It was when he retired that I became chief, which was 2005. But I've been involved in natural products, predominantly microbes, for a significant number of years, let's put it that way. Now, ask the question, why natural products? And both Wen and John referred a little bit to this. This is as of 2014, end of it. And the green, though not this ugly looking yellow as it turns out, the green are those materials that are natural products. Now these are all drugs. The total number of small molecule drugs was 1,211. That's approved worldwide, all diseases, and they're counted one time. So if Europe defines a drug and approves it, and then a year later the United States approves the same drug, it's only counted one time. If you look at what pharma, spelled P-H-A-R-M-A, says in the number of drugs that are in clinical trials, particularly in cancer, they're utterly in, they're using what one might say their statistics, because they count, and I've gone through their website many times. If you have one drug and 5 cancers, that's 5 drugs that are being checked. So divide by 200 or something like that to get some accurate figure. But the essential point here is that everything, I draw it along here, those are either natural products or are chemical kissing cousins of the natural products. If you look at cancer, and this now goes from 1940s, actually 1935 was probably the first year, this is the proportion of non-natural product. These are actually natural product-like synthetic molecules. ATP mimics in a lot of cases, or they fit into an ATP cleft. So you can see the vast majority are in fact natural products. Now, if you look, and I've done this from 1981, if you look, and I've just taken natural products now, plus the yellow. What are the yellow? Well, the yellow are what, over a beer I might tell you why we actually decided to use the S star. But in practice, those are modified nucleosides, modified adrenaline, etc., and so forth. There was some pushback from some of my chemical colleagues over calling them natural product derived, and in practice they are. Here's some very good examples. You may know, those of you who are physicians may well know of molecules such as ribavirin, which is also used as one of the antiviral agents. But it turns out that these 2 molecules down here are the ones that revolutionized antiviral and, for that matter, a fair amount of anti-cancer work, because these came from a sponge, and in the 1950s. Now, prior to the discovery of these two natural products, it was axiomatic that you could play games around the base of the 5 nucleotides or nucleosides, but you had no biological activity unless your sugar was deoxyribose or ribose. Well, if you look at this and you remember your chemistry, arabinose is merely an epimer of ribose. It turns out there's more arabinose around than ribose. That's beside the point. But until these compounds were found, they did not have any agents that were biologically active where the sugars were other than ribose or deoxyribose. The moment that these two were found, and I might add, though they were isolated from sponges, they've now been re-isolated from a microbe isolated from the original sponge, though I might add, 60 years later. And its microbial origin for spongothymidine and spongyuridine. So that's why we call them S stars. They really should be natural product derived. And this gives you some idea of what the molecules are that have descended or ascended from those, starting from vidarabine, where you now have ARA-A. ARA-C, I might add, was synthesized by a group that knew of the arabinose discovery. And ARA-C has now been found from a microbe as well. Azidithymidine is extremely well known. Well, if you can play games with the sugar, what happens if you cut the sugar? That gives you acyclovir. These are where you put a fluorine group on the sugar, and you end up with something that's quite well known. And then this one is the one that is your $80,000 drug treatment for HCV. In 6 weeks of treatment, you cure HCV. It actually is a masked nucleotide, though it isn't realized. And in fact, there was a very recent court case where Merck have now just received a significant sum of money from Gilead, because it turns out that some of this was from a Merck patent, which people did not know. However, let's look and see what happens when you play games with climate change. And you get carbon dioxide, evolution of carbon dioxide, high volumes of carbon dioxide. Well, as you know, plants basically use carbon dioxide and water to photosynthesize. I might add pseudoalgae. So anything that has a chlorophyll group in it or something akin to chlorophyll uses carbon dioxide. Well, if you start playing games with the amount of carbon dioxide that comes in, this is the normal flow. But you get all sorts of crazy things occurring. Because now when you increase or decrease the ratio of nitrogen to carbon dioxide, wherever you are, you start getting strange things occurring with plants. And this is not hypothetical, this has already occurred. So you can get decomposition of which you don't want, you get slow decomposition, which you may not want. Either of these can be dangerous, some of these can be useful. When you start getting into above ground work, you start getting some crazy things going on. And bear in mind that, as John mentioned, most of these are plants are a repository, if you will, or a depository for microbes, single-celled organisms. Plants do not exist in the absence of a single-celled organism. If you look at a plant, you've got single-celled organisms, fungi, protists, bacteria, in the rhizosphere, which is this area around the roots. You've got them in, on, and swimming in close formation on the leaves as well. So you have to think of a plant as an ecosystem. I might add that you get some other strange effects from other ecosystems, because methane is one of the rather nasty gases that is naturally produced that actually does act like carbon dioxide. It's a heat sink in the atmosphere. But what are the sources of it? Well, when you look at it, anthropogenic sources are a major thing. Those are predominantly cattle. and other ruminants. You get a fair amount coming, you get some from the organism, but wetlands themselves also produce it. Now, this is predominantly bacterial or fungal. And you can see in some of these where they have come from. So what you end up with is a problem. Normal living produces an agent that you really don't like, though you don't realize it. If you look at methane again, you've also got and people do not realize this, but if you go into what are known as the methane seeps, which are off the coast of North America, and actually they're off every coast, there's because of the physics of methane, and when you have pressure, you can actually crystallize methane as a hydrate. So you get a methane sink, which are off the coasts of the States and other major countries, where you've actually got what looks to be crystalline methane, which is methane coming from the normal microbial work, if you will, in the seabed. But it stays there as a crystal, and you can actually see these and use them. And the microbes that live on these methanogens are actually extremely useful in producing C1 compounds. And all people have to think of, anybody wearing nylon, it's due to a nice, happy little microbe doing some work originally for methane. And there was a very large amount of work done by, well, Cortels is the British company that did this, on methanogens and how to produce raw material to produce the polymer that is actually nylon and others. So methane is something that's not thought about, but it's a problem. Now, let's move a little to what can, we hope never does, but what can happen. This is a moon's eye view of Antarctica. And why do I say the Antarctic? Well, the Antarctic is the 6th continent. The Arctic is merely a sheet of ice. There is no such thing as the North Pole. The Antarctic, on the other hand, is a landmass covered in ice. But within that ice, you have the world's largest supply of fresh water. And in fact, if this area of the Antarctic ever melted, LA will now be a rather damp place. It'll be about 6 feet underwater. New York and Brooklyn, well, I live in Philadelphia, and I'm 180 feet up. So let's put it this way, I would have an oceanfront property if that melts. And this is one of the things that can happen. So this is an absolutely worst case scenario, but just gives you some idea of what people do not realize. But you can see the various and sundry options and things that occur. You've got invasive species going up with warm oceans. Well, Jonathan showed some of that, but in a different context. And now what happens when you get acidification? Well, you lose your seabirds. Calcified organisms go down. Fish die. Predators can go up and down depending upon whom they are. So all of this is a problem. If you look at the current Antarctic currents, and I make no apologies for the alliteration, you can see that if you do get some version of melting, you've got all sorts of nasty things occurring from the Antarctic that will affect everywhere. However, let's look at it from a slightly different perspective. This shows you the fresh water, which I talked about. I might add there are freshwater lakes that are a mile deep under the ice in Antarctica, and they are now being investigated from a microbiological perspective because they've been sealed systems. So it would be extremely interesting to find out what is there. But this is what happens, and let's move now to something that John alluded to. The coral reefs, and the one that everybody thinks about is the Great Barrier Reef. Well, last week I was actually on the Great Barrier Reef. I was in Brisbane at the bottom end of the Great Barrier Reef. It's 4,000 kilometers in length and goes right up to Papua New Guinea. And I've been there many times. Uncle Sam paid for a fair number of them in my days, and I've been there other times as well. But water has begun to rise in temperature. up to over 2.5 degrees centigrade. Now, this is from NOAA, and this just gives you an idea of what happens with coral bleaching. Now, what's meant by coral bleaching? Corals live effectively by photosynthesis, though you may not realize it. The coral obviously does not photosynthesize, but within the coral, there are single-celled and multi-celled organisms. Call them algae, and call them zooanthellae, depending upon the size. They actually produce the energy within a coral bed. But this gives you some idea of the danger that's going on. This is at the moment. This is not hypothetical. This is actual. And this is just what can happen if you start play some probabilities and look for the thermal stress in the next 3 months. So as you can see, it's already started. This is just an idea of algal evolution. This is under ideal conditions. However, if you increase the temperature or decrease the temperature, your ideal conditions are no longer there. So this is what it was. What it's going to be is a good question. However, this is what happens. The El Nino, which has been around now for 2.5 years in the Pacific, has caused the rise in sea temperature of approximately 2 degrees centigrade. Zooanthellae have voted with their feet. They're no longer there. This is what it should look like. This is what it does look like currently. You want 2 examples? You've got one in American Samoa, where you can now see the coral reef is now predominantly white. This is in New Caledonia. Now that's a staghead coral. That can take centuries to grow to that level. It's now white. Do they recover? Sometimes. Do I believe they will recover? Maybe 25% of the time. Well, this just gives you an idea of the surface temperatures around the world. And I might add that this center here is where Jonathan and I and Win and Luba were last year. This is Kazakhstan. Now, the normal temperatures there are plus 35 degrees C in the summer and minus 35 in the winter. I think you can probably add 5 degrees at both sides of that. And that is a desert. Well, it's a steppe, but it's basically a desert. So this gives you just some idea, and this gives you a scalar with color. Not happy. Why is there a problem with coral reefs and drugs and sources of interesting molecules? Well, one reason is this. This particular sponge, it's called, though it looks red, it's actually called a yellow morph. And molecules from this sponge have been looked at for many years. And there was always a debate, what produces the molecules? Now, bear in mind, NCI funded most of this work, and Gordon and I spent a lot of our professional lives working with these things, getting agents from marine invertebrates and others. And it turned out that you started looking and realized that it's quite possible that these are in fact produced by microbes in, on, or around the sponge. And work was originally done. Carol Bewley, who gave a talk, she and I gave a talk in this series about 3 years ago. Carol's across the road in Building 4. She was the one who was working on the thiopalamides back in her graduate school days, approximately 20, 25 years ago. And it was thought to be, is this possible? Well, by some absolutely beautiful work, this is almost Nobel level work, where they took the sponge, isolated every individual cell in that sponge and what was in it, on it, or around it. They don't ask me how many million cells in 96 well plates are actually is 1536s. They actually did. But the publication list on this has almost more authors than in the article. But they found that this particular compound, onamide, they found genes corresponding to the biosynthesis of this. And I might add that this part of the molecule, I have another set of slides that I gave in Brazil a few years ago, showing a blister beetle out of the Amazon wearing a scuba set. because this part of the molecule is pederin. And I lost it. This is pederin. Pederin is a toxin from the blister beetle, which we know is produced by a pseudomonad that is on the integument of the blister beetle. This comes from a shallow water sponge of Okinawa. So, having found that, they then proceeded to find an absolutely unknown microbe that has not been fermented yet, but it has had its complete genomic sequence obtained starting with one cell. PCR does work, ladies and gentlemen, but it takes a few years and a lot of graduate students. However, when they did this, they actually found that from that particular sponge, there were 32 at that time, this was 2014, there were 32 known agents that kill cancer cells, I'll call them that, not anti-cancer agents, but cells that, cytotoxic agents, 32 entirely different structures. 31 of them had their genomes or their genetic machinery in that particular microbe. What is even more fascinating is that the start of every one of those molecules was ribosomal peptides. And the best way I can describe it is how it works. For those of you with an agricultural background, excluding John, a disc harrow, which is the device that you tow behind a tractor to turn over the soil, and you have a series of discs. Well, think of an enzyme or enzymes that are a series of discs. You've got a ribosomal mediated peptide, and for this particular compound, disc 1, 5, and 7 operate. That's enzymes 1, 5, and 7. This one, enzymes 2, 4, 5, and 6. But it's the same ribosomal peptide. And this is a beautiful piece of work. And now it's being shown to be very, very common. So when you were taught that a lot of these were not ribosomal peptides, they actually are. Now, here's the point. Not all compounds are produced by microbe-invertebrate interactions, but I will go out on a limb and say probably 80% of them are, though I'd left the figure off here. What happens if you now go the other way and you now go from a very wet environment to a very dry environment? The Atacama Desert in South America is rather warm. Let's just leave it at that. And it has a chalk-like consistency. One of the chaps I know well actually started looking at the microbes that coexist on that. Effectively, these are dried out microbes. And they started finding some very interesting molecules when they did the Raman spectra. They actually found alga there. They were probably cyanophytes. But they then, because cytoneumin is in fact a cyanobacterial product, they call them algae, but they're actually cyanophytes, cyanobacteria. Well, now you start looking at the particular genome of those molecules that produce, and they start producing some very, very interesting and very strange peptidic-like molecules. This was a chemist that did this work, Journal of Organic Chemistry here, and it's actually from a group at University of Aberdeen. They also did the complete sequence. And again, the reason why this compound is a rather interesting one is this is what is known as a lasso peptide. It's a peptide that has a ring, but it then has a tail that loops back through the ring. So you can think of it as a knot. And these are very, very common as toxins. Plants, or is it the plant is a good question, produce a lot of these as insecticides. And actually some of them have antiviral activity. So this is what they started working on and discovered that they could fish the genes out. Again, you don't have to ferment the microbe. You end up with the gene. Now, if you now move to the plant area, what you find, and this is just this is basically a simple way of looking at it. It doesn't look simple, but it actually is. You've got endo and epiphytes. An endophyte is a microbe that is within the host, the plant, An epiphyte is something that is on the host. Now, when you start looking at these, this is now an idealized schematic of what you have between a plant cell here and a fungus here, which, as John showed in the bat example, the fungal, I call them epithelia, if you will, hyphae, go into the inside of the plant from the outside. And the bacterial cell is outside the plant, but then does chemistry and talks to the plant by chemistry. And I might add, that is very common in us as well. There's a comment I often use, the question is, how human are you? Well, the answer, depending upon how you wish to define it, is somewhere between 10 and 15, 18%. That's when you count the number of cells, number of single cells in the body versus the number of mammalian cells that compose the body. And the answer is less than 20%. So that's the microbiome. Well, this is the equivalent in a plant. And you have chemistry talking between the microbes. Well, here you've got chemistry going back and forth between the plant, the fungus, and bacteria. And if you start interfering with this, you then have a very unhappy plant. And if that plant happens to be a foodstuff, then you fall into exactly the situation Jonathan was talking about with food security. And you don't know what's going to happen. But what is even more intriguing is that these interactions produce compounds that have been known, compounds of abuse and compounds of use. Note the middle one here. If you make a diethylamide of that, that's LSD. And these are very well-known products that the ergot of rye, which is an epiphytic infection of the rye plant with a fungus, Claviceps purpurea. And there's an argument that the witches of Salem were due to the hallucinogenic effects of these particular molecules on rye that had become infected with ergot. And there's also another one. It's also a version of this is used to reduce prolactin secretion in both men and women, to bring down the levels of prolactin. Therefore, if you do that, women who could not conceive can, because they don't ovulate in the presence of very high levels of prolactin. And so 5-bromoergocryptine came from this molecule, came from this particular fungus as well. You lose that fungus, you both lose your hallucinogenic effects, but you also lose some other rather interesting effects. And when you start looking at this, I always joke that this is the sort of work that tells you that Lysenko may have been correct. If you remember your genetics, Mendelian genetics is one, but Stalin and Lysenko claimed that you could have induced traits that were then transferred vertically. Well, it so happens these are transferred vertically by the plant. These microbes actually are part of the seed. And what you end up with, Claviceps being a beautiful example, and even better, the morning glory, there is a microbe that is part of the seed within the seed itself that then carries upwards as you get a new generation. The self-same thing happens, I might add, with another very well-known compound in our field, which is bryostatin from Bugula neratina. You have two types of Bugula, those that contain an endophytic bacterium and those that don't. The ones that do, the larvae carry the bacteria to the next generation. The ones that don't, you simply have Bugula. So there is a possibility that some of the Lysenkoisms may have been correct, but we know it now. Now, of course, if I say that in a school audience, I get howled off the stage or dragged off and tarred and feathered. Here I could say it. Now, you just get some other examples. The odor from plants or flowers that you like are often due to these molecules known as terpenes. They are usually produced by microbial components of the flower. So one might say, oh, well, the flower looks nice, but it's lost its smell or its odor. One of the reasons being a loss of bacteria. How do you find microbes? You know, we've been saying, I keep saying we're losing microbes. Well, how do you find them? Classic method is fermentation. And I spent something like 25 years of my professional life trying to do this, sometimes succeeding, sometimes not. Today, what you do is you hire your local gene jockey. And in addition to your local gene jockey, you also hire someone who knows their way around a computer. Because effectively what you do is you get the gene clusters, so you don't have to ferment them anymore. Just have the microbe, get your gene cluster, okay, you have, and then you can idealize it. You can use various and sundry techniques known only to those people who gaze into it. Used to be cathode ray tubes, but now it's an LCD screen, and come up with ways of playing games. And you end up with molecules or the genes that allow you to find molecules you didn't even know existed. Here's an example, just a simple one. Well, what you can do is you now have, and this, by the way, I took this from the slide and realized that they had made an error. This should be BGC for biogenetic gene clusters. And everywhere else they talk about biosynthetic gene clusters, but they obviously, I'm going to have to, I know Fishbach quite well. I'm going to have to kid him next time I tell him that he needs to have his graduate student do better proofreading. However, this is what you can do. You can now group these using computerized techniques, and you now can come up with, okay, this is actually a molecule that produces rifamycin. Now, those of you who remember, that was one of the first drugs ever used against TB, and it still is used. So you can find these without even fermenting. Well, now let's ask a different question. You're finding these today. Well, if you get a rise in temperature and it's coming from a marine-based microbe and it's part of a coral reef, it's no longer there. So you don't even know A, what's there, or B, what you're missing. This is where the computer jocks go crazy. They take the complete biogenetic sequences and proceed to cluster them. And in fact, I've seen this done and I didn't believe it. I do now. You can actually do this with a consortium of microbes. And this is actually how a lot of the microbiome has been done. And you put them into clusters depending upon what genes you have there. Then when you find another gene or gene cluster, you can say, oh, it corresponds to this one. Can't use this, sorry. It corresponds to this one. And this one I know contains genes that produce A, B, C, and D. Therefore, I've got something that is related to A, B, C, and D. So this is what's being done now, rather than the way I spent my life trying to ferment the little buggers and failing miserably, or only getting one compound. And bear in mind that about anywhere from 5 to 15% is the highest I've seen of the genes in microbes actually have the capability of producing molecules. The problem is switching them on. Again, we don't know what we're losing. This is an example of where you've got interplay now between a termite, fungi, and bacteria. And what you found from this is just a termite mound. You can see the little critters here. What you find is that you get some very, very interesting molecules. Some of them I spent an awful lot of time and money at Uncle Sam. We grew over 3 kilograms of this particular molecule, because that was the precursor for 17-AAG, which we took through to with help to phase 3 clinical trials and variations on it. But it's produced by the same microbe that produces this. Well, we knew of this. We had no idea of this, nor of this one. And a couple of these, we'd seen others, but we didn't know. So now what happens is that by looking at the interplay between microbes, insects, and others, you start seeing molecules that you didn't even know existed. And in fact, this comes back to a point Jonathan made about symbiosis and the interplay between molecules. One of the things that I did before I retired, a few years before I retired, is we started doing some playing up at Frederick, where we took fungi and bacteria that had never seen each other and proceeded to grow them together. And we started getting molecules switched on that we didn't even know existed. Now, we didn't find the genes, we looked for the molecules chemically. So there's an enormous number of potential molecules out there. And this is one of Wynn's comments about in silico. My answer is that you're not going to find a damn thing in silico that you don't already know about. Whereas if you look at them using modern genomic techniques and modern ways of playing, using in silico, but looking now not at the molecules themselves, but at the clusters, you now realize that there's an enormous number of molecules out there. Just to give an example, in one fungus, there are over 75 different clusters identified. And that does not include terpene synthases, which have at least another 20. So you're looking around 100 different genomic clusters that have the potential to produce molecules we've never seen before. Or in some cases, as in this case, they produce molecules we haven't seen, and molecules we already know about. And that's basically my take-home message, which is because we don't know what's there to begin with, we don't know what we're losing, but we know that we are losing. And just this was the slide that Gordon and I put together many years ago. This shows just some of the materials that we played with at the Natural Products branch. The background here is in fact taxus bark, and that's Taxol. This, no woman in the audience ever wants to meet face to face, doxorubicin. But this is the streptomycete that produces it. This is wart mannan, which is produced by this fungus. And a modification at this point, that's now in phase 2 clinical trials. And this delightful looking little creature, I have a lovely story about this one. I was giving a talk at Cold Spring Harbor, Woods Hole, Cold Spring Harbor, many years ago. And I was asked, what does NCI collect? So I said, well, if it's brightly colored, slow moving, has no obvious means of defense, and I added, is fatty and over 40, and thank God she didn't put the over 40 in the article, because what I'm describing is this little creature. Now, these range in size from about my fingernail to about 35 centimeters and weigh up to a kilogram. These are nudibranchs. They are a shellless mollusk, so think of them as a snail without the shell. But nothing touches them, and that's breakfast for anything up the food chain. The reason being that they nibble, eat, ingest, is whichever verb you wish, molecules from usually cyanophytes, cyanobacteria, and or other bacteria. And the toxins are then put onto their backs so that anything comes along and goes chomp, it's chomp, die. And though the little creature gets a little bit irritated, the predator is forepaws up, or its fins are up and it's floating to the surface. Those are organisms we like to look at. We now know, some of you may remember when dollar-statins were put into clinical trials at NCI and other places. They came from an organism like this. We now know that they're actually produced by a blue-green alga, a cyanophyte, that the host organism, which in that case was Dolabella auricularia, eats. And the toxins, and there are about 25 of them, are in the fat layer of the organism. So, as I said, we don't even know what's there. But if this little organism gets either too cold or too hot, you're not going to be able to find them. So that's basically the story. So anybody wants to throw brickbats, rotten tomatoes? Josh will from the back. Particularly when I tell him that a plant is nothing more than the host. He's a botanist. So many thanks indeed.
Oh, thank you very much. Thank you, John. So I hope we have some questions or.
Very simple.
Yeah.
What is the main? In methane, is the main reason it's such a bad gas is the heat capacity?
Yes.
Or other photophysical effects?
It's basically the heat capacity and the fact it's highly volatile. So it'll sit in the troposphere. And it's actually much worse than CO2.
One other question. In your ergot examples, is there any change in the distribution with climate?
There could well be, because ergot is a fungus that is a spore former. And so depending upon winds, and what I didn't put on any of those was the increase in wind velocity in certain areas, you're going to get spreading. You can get, so one doesn't know. I use that just as an example.
But there's no known change in ergotism and old-fashioned things like that?
I do not know. I haven't looked, John. No, I mean, there could be an awful lot of this, but people don't look for this. I looked at it as a chemist, trying to find ergot, trying to find the molecules back in my youth. But how they came about was only learned later. Go on, John. Go on, Josh.
We need to use the microphone.
You won't?
No, I won't. But I will ask you, in all honesty, are there any groups of compounds yet that are still produced, to our knowledge, only by plants?
By plants? Yes, there are. And
Some of the
off the top
alkaloids?
of my head, I cannot, not all of alkaloids, no. Some of them are alkaloidal, but there are alkaloids actually produced by fungi that come from the plants. Right. I know I wrote this in one of my more recent articles, but I'm blanking on which it is. When they removed the bacterium from the plant, the other molecules were still produced by the plant. So yes, there are molecules that are plant. There are other molecules that may well be produced by an interplay between the plant and the agents. But there are molecules such as maytansin, which originally was considered to be, and Gordon and I actually published this in a chapter with Floss, where it was thought that a precursor to maytansin, which is antimitosin P3, was known to be produced by an actinomycete in the rhizosphere, in the roots, and that the plant then did a transesterification to produce maytansin. A year and a half ago, a group discovered that no, there was actually a bacterium in addition to the antimitosin P3 producer that did that transesterification. And it does not occur in the plant at all. The plant absorbs it and uses it as a protective agent. So as we learn more, the paradigm shift.
Just so related to that, one quick follow-up. Where are we now with Taxol and the fungi?
Now there's a very, very interesting story on that. There's a paper that just came out, and in fact, are you still an ASP member? Okay, because one of the things I do is I write an article, a fun article for me, every quarter, and I've just used this as an example. What happens in the yew tree when it buds, or basically you get a twig, a branch coming off, the bark cracks. And a couple of people in Canada did a beautiful piece of work where they looked at the fungi, really the microbiome of that area, and discovered that within Taxus there was a fungus that was an endophyte of Taxus. When they looked hard, they discovered that Taxol was within the endophyte, but in a lipid layer. So you can think of it as being little lipid droplets. And when there was a crack, you got a fungus coming in that was a lignin hydrolyzing fungus. That Taxol, they laid down basically a death wire. Cross this and you're dead. Because Taxol was originally, and still is, a very good antifungal agent. So what happened was, it killed it. So they fished out that that fungus, and discovered that when they fermented it outside, if they put methyl chloride, which is a breakdown product of lignin degradation, in there, the level of Taxol production went like that. Now, is Taxol a plant product, a fungal product, or are the two roots to Taxol? That's a question that is not yet definitively answered.
So,
Go on.
I'm sorry. Jonathan, maybe we could ask you, in this part of the world, particularly, we talked about this before, but maybe you'd say a word about it. We keep hearing on the radio that there's a epidemic of Lyme disease permeating around in the central Atlantic coast. And we all have deer wandering around. We don't know whether they're the culprits or what can be done. And then there's this whole business of great concern about chronic chronic Lyme disease, and many articles now about whether people should be treated for 2 years with antibiotics and whatnot. Now, looking upon this from the perspective of wild animal, how would you fit an approach to the one habitat or concept that you advanced in dealing with, say, this issue of, just to put it locally, or it's more than local, but of the Lyme disease and Borrelia?
Yeah, so Lyme disease is certainly a very interesting example, and I didn't touch upon it in my presentation, but basically one of the factors we believe why we're seeing a high incidence of Lyme disease is the way that we live in these types of suburban environments. And you see, you've got homes very close to these forested areas, and it's caused a lot of fragmentation of these forests, so we have a lot of edge on these forests, and the research has shown that you get a higher abundance of the mouse, that's the main reservoir for the Borrelia organism, the white-footed mouse, in these fragmented forests versus more pristine forests. So these pristine forests have a higher biodiversity of rodents, whereas these fragmented forests have mostly a single species white-footed mouse. So one of the theories is that what we're seeing is the loss of that dilution of, so we've got a tick seeking a host. If there's only one species, the competent reservoir, then it's going to find that host and get infected. Whereas that tick is in a forest with other species that aren't competent reservoirs, then the probability of it becoming an infected tick decreases or declines. So I think that's one factor is the way that we live in our suburban environments. The other factor is these suburban environments are perfect habitat for deer. You probably all know this, right? It's impossible to grow a garden in this part of the world without deer eating it. And so you've got a very high prevalence or high densities of deer. You've got higher densities of ticks. And so that's probably another factor. But I think the main factor is the loss of the dilution effect from biodiversity. Probably would have been a good example to use. It's also actually being impacted by climate change. Lyme disease is creeping further north into Canada, Ontario. We think migratory birds are kind of taking the ticks and dropping them, and then again they're able to survive at these higher latitudes. What to do about it is a big question that's being discussed right now. There's a number of techniques that are being used. When I was in Virginia, in the Department of Game, one of the things that they're starting to use were these stalls that deer would come in and get treated with pesticides to try and treat them for ticks. Obviously, culling deer and keeping populations down is important. But that's not easy in these suburban environments where guns and people don't usually mix very well. There's a lot of discussion about modifying habitat to try and prevent ticks from coming into people's yards. I think I think for me, we need to get some creative solutions to these problems. One of the areas of research for us, as I mentioned, is development of vaccines to vaccinate upstream. Can we develop a vaccine where we can put in an oral bait, where we can put in these forest areas which can vaccinate the mice against Lyme disease to prevent that transmission? So I think right now you're stuck with DEET and checking yourself for ticks after you come in from the yard. But I think there may be some creative solutions in the future.
Yeah, that was the dilution effect was actually mentioned a couple of years ago at a meeting I was at in DC, where it was actually your group wasn't there. This was EPA, amusingly. We're talking about the fragmentation and the lack of dilution. A similar thing has happened in Colorado, in the Four Corners area with Hunter virus, where you got changes there, and now you had much more in the way of mice coming in to feed on grains that happened to be in the houses. So you had, and these were areas where young students would live. Hence you had this apparent infection of youngsters, not guys my age, because I wasn't going to live in the outback. They would. And that was hantavirus. I should add as well that John and I were 30 years apart in the primary school. Although I understand my face was on the wall with "Don't know this guy." You know?
Well, listen, I want to thank you both for very, very informative, very interesting.
Thank you.